What is going on? It is Sunday, February 20th, 2022, and February is over one third done. Yeah, it's going super fast. What can I say? Time is flying, and it's been fairly productive. I've started up the series of videos on the on biology, cell biology, and in general anatomy, and I've switched over from the study of neurons, just if you remember, and my video yesterday had to do with junctions. So I was talking about gap junctions specifically, but I had made reference to other types of cell-to-cell junctions and cell-to-basal lamina junctions that are involved in keeping epithelial cells and other cells that need to talk to each other communicating and connected, and also stabilized, but yet flexibly stabilized on other parts of the body and other membranes and things like that. So those are important at that point to keep in mind. And there's a lot more to go into regarding gap junction, all the junctions. Really, they're fairly complex in their actual chemical interworkings, which we may need to understand in order to understand how eternal life can be achieved, right? And their role in longevity and health span, and pretty much everything related to cellular health, right? So, and the body's health, obviously, right? But for now, I'm going to take a detour into some even more basic territory. And I think it's important to go and cover all of this. Is something that I really want to delve into because it's important, because it's at the fundamentals of everything cellularly we are talking about. We will go back to study of the neuroanatomy at some point, because again, that's as far as longevity is concerned, it's integral and crucial to understand how that works. But it's also crucial to understand how this works, and this is actually the foundation of that. So you've learned about all this in high school. Again, a lot of these topics you've learned about, I'm sure, in some measure in high school, and even before that, in like 7th grade, 8th grade biology in the United States, I believe they teach you cell phases and cell cycles and whatnot. Whether you're capable of really appreciating or understanding the magnitude of what they're teaching you at that point varies from student to student, but at least for me now, is really important to understand all of this, especially considering the study that I've taken up, the company that I'm working on forming, and the research that I want to move into. It's really, this is the core understanding of life and how life sustains itself, specifically human life and all life really, or most life. There's obviously life that is less complex than us, that has been sustaining itself for eons and eons through mechanisms that are analogous but different than the cellular mechanisms that support us. But they're all worth studying, honestly, and it's all interesting to me personally. So we will talk about, back to the topic at hand, we are going to talk about cell division. We're going to continue to go through all of the cell processes, and this is like fundamental to cell biology, cell division, right? Which is something that it varies from cell to cell, right? So let's start with the first. Let's start with the overview. So in a cell's life cycle, you have two phases. There's two globally large-scale phases of a cell's life cycle. The phase is the phase that the cell spends the most of its time in. The large overarching phase is the interphase, and the phase after interphase, which is the much shorter time that the cell spends dividing and duplicating itself into two new cells or multiple new cells, whatever, is called mitosis, right? And so you have interphase and mitosis. Interphase can be broken down into smaller phases, as some of this is definitely review. but it's something to keep in mind. Interphase can be broken down into several smaller phases. The first major one of those to keep in mind the is the cell growth phase. It is the first cell growth phase, called G1. So the G1 phase is where everything is essentially your cells are duplicating their internal structures. They're duplicating their organelles. They're duplicating a number of things. Right? They're essentially forming separate units. I think it's best to outline what goes on in the G1 phase, because the G1 phase is, and it's always good to go back to wiki. The G1 phase is, what's interesting is a very variant length phase. Here we go. The G1 phase, the cell grows physically, increases the volume of both protein and organelles. So it's making more organelles, it's making more proteins that are sometimes specific and also generalized to the cell. Right, so again, all that is happening, and this can be happening for as short as a few hours to as long as years, and it depends on the type of cell that it's happening to. In the G1 phase, we have this time spent because we have different kinds of cells in our body that have different growth rates and they need to regenerate faster. So in the stable phase, in the division of our cell types according to their regeneration cycles, we have the fastest growing ones are the labile cells, and those are in certain epithelial areas of your body, right? In your mouth, on your skin, you have cells that are, your skin cells are essentially labile because they're always falling off and dying and growing, etc. And in your epithelium, in your mucosal tracts, in your urogenital tracts, in your stomach lining, these cells are constantly getting regenerated and repaired and dying off and getting regenerated and repaired and being exposed to cellular trauma, which they're meant to do because they're designed to protect you from the environment and environmental issues. So they need to suffer damage and they need to grow back quickly. So the next type of cell is a lot higher, that has a longer interphase. Has a longer G1 phase, which is essentially equal to a longer interphase, is stable cells, and we'll go into stable cells a little later on. They're obviously an entire topic, and they depend on their different organs have stable cells. The liver is the most well-known organ that has a stable cells structure. The liver can regenerate as necessary. So think of stable tissues as they don't proliferate constantly, they proliferate as needed, right? So these cells are capable of dividing in response to injury or loss of tissue mass, right? So again, your liver cell is considered, I think 40% of, if you take away 40% of your liver, your body can function and the liver can regenerate itself regenerate that 40%. It's a very robust organ in that sense, and the stable tissue, it's because for that reason it's counted in the stable tissue. Stable cells constitute the parenchyma of most solid tissues such as liver, kidneys, and pancreas, including endothelial cells, fibroblasts, and smooth muscle cells. Each proliferation of the cells is particularly important in wound healing, so reactive cell division, right? And then finally, going back, we have your permanent cells. And your permanent cells include those cells which pretty much almost never, practically never, virtually never leave the G1 phase. They're constantly active, and they may be duplicating organelles, but they're never getting to the synthesis phase where they're doing the S phase, which they are doing DNA replication, because they're never intending to actually divide. They're essentially quiescent or senescent. They're more permanently quiescent or senescent in that sense. And remember, those terms are very important because quiescent talks about cells that never that temporarily go into a quiet stage. And senescent means cells that are permanently in a quiet, inactive in terms of cell division stage of their lives. So yeah, so these permanent cells examples are your neurons, your muscular, skeletal muscular cells, many other, actually a number of other types, but those are the most prominent ones because you've heard that your brain cells never actually divide, and that's a reflection of the statement of them being permanent cells. Okay, so your more proliferative cells are again are essentially cells that are epithelial in nature, and you have these. Just this diagram is kind of like depicting the difference between different cell proliferation strategies, or their cell growth, cell division, cell proliferation, the different views of the increase in the number of cells as they're dividing, etc. So, and then another example. So, in cell division, and this is kind of like, you've seen animations of cell division, I'm sure. What's happening is essentially in these animations is a combination of phases is occurring, right? I don't know if we're going to be able to see. Yeah, we can see generally what we're going to be talking about. So you have the different components, which I'm not going over all just today, but I'll just maybe briefly mention. You have your centrioles, you have your mitotic spindles, you have the DNA that's migrated in this line and that's being separated by these centrioles and mitotic spindles, the duplication of the cytoplasm, and the cell increasing and splitting, and at some point the cytoplasm is cleaved, and there's two nuclei that are formed. So that's the general gist of cell division, right? And so I'll keep it at that. Next time we'll be talking more about DNA replication, and briefly touching on DNA replication. We'll get into the nitty-gritties of DNA replication a few lectures later, right? But we'll talk more about like S phase, and then the G2 phase, which is the secondary gap phase, which is what happens right before cell division. Essentially, both of those phases are important. All of these phases are important. So that's it for today. Hope you enjoyed this video, and I hope you found it helpful. I'll see you later. Subscribe and like to Sam Q Scientific, and have a nice day.